I will start sharing my screen now. That's the screen here. So I will start sharing.
So, Sabarna,
Yeah.
we are live now. So, your lecture is start live streaming on YouTube as well. Dr. Ishan is able to
Okay.
see the problem.
Okay. Okay. Perfect. Okay. So as I was mentioning earlier, that the session I have divided into three parts. So the first session will be the basics. So firstly, RStudio, according to me, after using for like 4 years, and I have learned RStudio on a time, I mean, that might be relevant to the participants here, when ChatGPT wasn't available. So I have kind of learned RStudio in the hard way, just scribbling through the codes, scribbling through the commands, and scribbling through the literature, etc. And I still believe that it is a very user-friendly programming language, especially for biologists and researchers who pursue biology, because I think it is super friendly in the sense that the complexities of the code are lesser than other programming languages. That's one of my personal opinions on RStudio. Now, the basic stuff here it begins is that, so R is the name of the language. I don't know if I can use my pointer or not. I think I can. So R is the name of the language, and RStudio is the platform where we implement R, or where we write the commands for R to visualize our data or to analyze data, etc. But it is better that the participants who are here know the actual meaning of the two terms, that please don't confuse in between. R is different and RStudio is different. People, the participants, they can write me later on during the session breaks. If somebody is there with a Linux computer or with a Linux system, sometimes Linux works on something that is called RStudio browser. So that is completely different. So these terms are really important before we go to the major stuff in the programming. So I have attached example screen here, so you can see that where or how we do the stuff. So once I finish explaining the slides, I will share my screen again with my own RStudio screen, and then we can start analyzing the relevant stuff for this first session. So the first thing is that here is the place where we write the commands, where you write your commands to analyze your data. Here is the place which shows the environment where the various variables or various formats of R are stored, or sometimes your output variables as well. And this is the console. Console shows what kind of programs are running within your RStudio environment, and this is the output, especially with the plotting and etc. So essentially, there are some more stuff which I will handle in a much more detail once I start sharing the actual way to do stuff for relevant to session 1. So there are something called R files, yeah. So as you can see here, this, wherever it is written, this is called an R script. So there are many. There are R script, there is R markup, there is R session, many. There are many ways to write commands or write codes in R. And this is one of the simplest way, because other session types like R markup, let's say, these has some tedious terminologies and tedious way to write the syntax, which most of the time can result in errors. So R script is the can be said the most easier way to proceed with R commands. So I think most of you might have knowledge of people having a knowledge of R might know that R operates through packages, and inside the packages there are functions. And before you use a particular function, you need to install those packages individually. So for instance, there is the very famous ggplot2. I'll come into to this in a moment. Then we have VCTRs, that is the R vector package. Then you have R lang package, and many. I think there is something called the very famous one that is called the tidyverse, which we will also use. There is something called tibble. Tibble is mostly for table making, and particularly when you need to mutate your table. So for instance, that you have an output that is generated using a set of analysis, set of data analysis, but you still need to insert an additional column. So for those things, something called as data manipulation, although don't take it literal in the literal sense, manipulation in the sense that you are trying to add extra variables or extra information to your data. That's only called manipulation in this aspect, actually. So the most important or the most symbolic packages that are used mostly in RStudio in terms of a biologist's viewpoint is that we use tidyverse, we use shiny, we use tibble, these are some of the majorly used packages. So another thing that is a kind of a boon, kind of a blessing to each biologist, that biologist particularly, that I think, as you all know by now, that I am a plant scientist, and in most of the cases before we analyze data, it is always in an Excel sheet. And here comes its user friendliness, that RStudio and Excel go hand in hand very well. It's a very compatible situation where RStudio and Excel go really nicely. Obviously, there are put data frames or let's say sheets into an R environment. I'll show you once I start doing the session on basics with you. And but RStudio is also friendly with TXT files, the text files, the text outputs. That is one of its great compatibility. So let's discuss a little bit about the basic data structure that is involved and that we might use in the coming sessions. And people who are taking this course and who will face real life challenges where R will be used rigorously, then I think these kind of data types will be really handy. So the first is that something called a vector, where you have these the numeric numbers. Then you have data frame, where you have two kinds. So X and Y. So you are depicting a kind of an information, which is a preparation for doing a graph. So you see that you have mango here, you have orange here, you have banana here, you have grapes here. And I think it's a mixture. So you can either put up to decimals, or you can round up the decimals. Like here it is shown 5.01, sometimes it's shown 3, 2.3, 12.3, etc. And R will take it very easily. And data frame, as you see here, this is one of the most common ways to approach or to use R analysis. So putting every result of yours into a Excel sheet that is already prepared in a data frame for R, and that is very easy to import it into R and then carry on with the subsequent thing that is called matrix. I think if you have some knowledge of programming languages before, matrix is something which is only numeric. So there is no data description. There is no space for data description, and that's why it makes it more tricky to use as compared to a data frame, where you see that you have a data description. That when you put this data frame into a function, then you can easily specify which axis will be your X, will be your Y axis. But that specification, that becomes really tricky to make. I mean, tricky to make in case of a matrix, because a matrix only has numeric information. And also we have the list, where you have a mixture of everything. You can have vectors, you can have a data frame, you can have a matrix in it, you can have text forms with also. I think lists are mostly, these are output of certain classes of our functions where you get a list. I think we will do something called a t-test in the intermediate session. I think that is also a list output. So you will get to know what is a list output in that case, that we will see very soon. So the plan for this session, at least that I have planned it so far, is that first we will see that the data loading into the R environment. Then we will see a basic interpretation. Third, we will use something called ggpub-r. So as I mentioned in the earlier slides, there is a package called as ggplot2, right? Package very universally used by biologists mostly. That means ggplot2 publication ready. That's the abbreviation for it. And most of the times that this is the most relevant way to create a graph. Yeah. And then, of course, we will learn a little bit about bar plots, the box plots, and I will also explain to you the difference between box and bar plots. And in general, we will talk about graphical visualization. The plans that we will do for the first session at least. But before I go into the complexity of the R environment, just a free discussion on of understanding statistical analysis when you have biological data. So the first thing that you can see here is essentially the bar plot with a standard error. That's the upper standard error bar. And basically it shows the effect of two variables, that is lane. This I think is the lane and one is the dose. And you have it grouped over two kinds of supplements. One is OJ, one is VC, and you can see already a colorful bar plot on your screen. Yeah. So this basically shows, so everyone knows, I think, so here is the mean of the a value that has been shown, that the mean of this OJ here at a dose of 0.5 is close to, let's say this is close to 12 or 13, let's say. And it has a upper standard error or upper standard deviation of around, let's say 6 or 7. It's only that this is a way of plotting the standard error bar, that either you can choose to plot the upper part, or also you can choose to show the lower part. That is also there. In most of the cases, I think in this example, it is shown in the both ways. It's both the upper bar and the lower bar as well. But it's a choice. I mean, mostly people go with the upper bar also certain times when it is not that important to show all the aspects of the standard deviation. The second thing, before we move on to the actual analysis part, is something called very, very important, that is called jitter. Jitter. Jitter is essentially that you are trying to show the distribution of your data points. Okay, so let's consider this bar. So this jitter, how can this jitter be important for the readers, let's say, so let's say this graph is published in a journal, and how this graph can be essential to a reader. So the reader can understand that, okay, 1, 2, 3, 4, 5. So this part is kind of clustered together, and this part is more clustered together, the data points. Whether this gentleman here, it has scattered a bit. So this important information, I think from 2024 onwards, this has been mentioned in most of the top journals, the top tier journals. I'm talking about Nature Communications, PNS, etc. So you need to show something like this, because the reviewer would like to, as he doesn't have the time to look through your supplementary Excel sheets. So they wanted that you plot the distribution of your contributing data set within the bar itself. Yeah. So that is a way to efficiently explain your scientific data. And of course, that also can be done with a box plot. And talking about box plots, that be very careful that a box plot slightly differs from a bar plot. A bar plot usually shows the mean. A box plot shows the median here, and there are many other depictions of the measures of the central tendency. So essentially, either it's a box, either it's a box bar. You are showing measures of the central tendency. So the measure of the central tendency that is shown usually by a box plot, that is the median that is shown, and also there is information regarding the quartile. So you have Q1, you have Q3, and also there are interquartile range, and it also shows the outliers. But never confuse a box plot with a bar plot. A box plot, these lines don't represent errors, okay? So this is a kind of a mistake that some people can do when they are new to this subject, that box and bar are, although they might sound similar, but they show completely different information. And that's one of the really important stuff in statistical biology, especially when you are trying to represent your results. One more slide before we move to the actual session. So a very relevant point of information for you, everyone who is trying to learn R, that R colors are different from Excel colors, okay? And there are huge varieties of the colors that are applied in a RStudio environment. And sometimes it is considered to be a good situation. Sometimes it's considered that it's very tedious, and you have to be careful in putting these terms together. So if you do a syntax error here, and then of course your command will not function properly. So same for other very complicated names, no? So something like Chartreuse, or like this big name like Cornflower Blue, or something like, let's say, you have dark slate gray and all this. So if you are using these colors, I mean, as a person like me, I'll be happy to use red, blue, and green. So, but yeah, so if you have very complex data set where you think that you can divide it by colors, then of course RStudio is a blessing to you, and you can go ahead and do that. I mean, of course, this talk will be recorded, but yes, this is a actual depiction of the colors that are available in art. So somebody who sees this video later point might find this as a reference point to choose for colors. Okay, so I think at this point I will stop my screen presentation sharing, and then I will share my RStudio screen very soon.
Before that, can you please let us, our participant, know how to download R?
Okay, sure, sir.
So just a quick lesson so that the person, those who are new to it, so they can easily download it. Or you can provide a separate sheet. We will share it with our participant. It's up to you, sir.
Okay, sir. I will see. I will maybe do it right now only, but yes. So, yeah. So, I will share my screen again. Okay. So, I think you can see my Google screen here.
Yes, now it's visible.
Yeah. So so basically, people who want to download RStudio, they can write here. First, they have to write R download. Now, here is a very tricky thing. This is something really tricky to understand. So the moment you write R download for Windows, then you have two options, 32-bit and 64-bit. So this is something very internal to your using, and sometimes it's very confusing. Sometimes 64-bit also, that if you download it on your 32-bit, it also sometimes works, and vice versa. So it's a very complicated thing, but and this thing, this whether your computer is 32 bits or your computer is 64 bits, you can look at it in your control panel and go to settings. I think it's about the system itself, and there you can take a look. But sometimes it can end up that you are doing trial and error. So you install both, and whichever is the best, you take that. So for me, I think it's 64 bits. So I will directly go to this link. So the first thing is that in this era, there are many spam this very scammed websites, or let's say fake websites. Okay. So the thing that you can trust here is the cran.rproject.org base. This is the website that you need to go to in order to download R. So you can go here. I think it is opening. So you can see it here that the first, this thing is R 4.5.1 for Windows, and it has some explanations, and then I would directly click on it and you get it, yeah, on your computer. Double click that and install it on your computer. So that was firstly for spoke to you in the couple of slides back, difference between R and RStudio. Downloading R does not mean that you have downloaded RStudio, because these two things are completely different. So you click here. This is called POSIT. That is the official repository for RStudio now. Here is the thing. So it's called Download RStudio Desktop for Windows. Yeah, just click on it and then you can start doing it. And it is already written that RStudio requires R 3.6.0 plus. This actually means that so 3.6 is a much older version. I think, let me guess, it is maybe 3 years back or 4 years back. So something which is some R version that is more than 4 years old should be the target actually. So I think in our case, I think that was 4 point something already that I showed you on the Google search. But yes, something recent version of R is sometimes needed to ensure that your RStudio is also working really, really smoothly. So this is something really important. Okay, so I think that was the few stuff about initial stuff about the how to install RStudio itself. Just a second. Yeah. So I think with that said, let's get on to the RStudio itself. I will share my RStudio screen and we will do some relevant data analysis together that is relevant for this first session. So I'll do that again.
Thank you, Sabarna. Thank you.
No problem. So I will go to this one. Participants, my computer is a little bit slow, so please bear with me. Presentation takes a little bit time to load up. Yeah, thanks, sir. So it takes a little bit time to load up. Okay. So I think we are now ready with my screen. So as we explained or as we discussed before, that this is the place where you do Art Studio. I mean, you put the commands, you analyze data, you visualize data, etc. But firstly, because I think there are people who are beginners. But firstly, because I think there are people who are beginners. First, sometimes it is necessary to look like you are doing something related to coding. So you can go to global options, and that is available in. So I have something called a GOB, that stands for Goblin, and you have also something called Dracula. So these are all the dark appearance settings which you can use, and if you are doing that in front of people or in a library, you can easily use that to at least let people know that you are doing something related to computer science. So I have done my Goblin stuff, which I really like this kind of appearance. So, so first we will try to import the data set, which I have already done to save time. So I have something called a dummy data. So just a little bit of information about that. So here is the portion where the data is stored. So here you can see, I think it should be somewhere here. Dummy. Or maybe it's somewhere here. Anyway, when I call this, you can know what I mean. So if you click here, this is called import data set. So what does it say? From text again, from Excel, from SPSS, from SAS, and from Stata. These are certain statistical application from where you can retrieve your data, basically. Because if you ask a biologist is just a layman in this aspect. I mean, a biologist is not a statistician. So let's focus on something which is very relevant for a biologist. So we just click on from Excel and a pop-up box will appear. So it shows you here that it is preparing import from Excel, and it needs a file URL, and then it will show you a preview of your data. And if you click on import, it will import your data. There is nothing else to be done at this point. And because I have already called the dummy data that was there. The second important thing is that let me write it down already on my screen so that you guys can know what is happening. So if I write this, so it says get WD. So WD is something really, really important for our analysis. But its importance comes a little bit later when you are asking Art Studio to find your file, to bring it to the session, and then analyze it. This, unfortunately, fortunately, let's say, we don't do it for the basic stuff. I mean, mostly this is used when you are really doing large data analysis. That by that I mean genomic data analysis, maybe RNA-seq data analysis. From there, I think you can, if you set the working directory at the start, then you are making the task for R a little bit better, a little bit more easier, so to say. But I think for the time being, you can always directly go to import data set. If you click on the type of data that you want to import, and you know where the data is saved. It could be saved in the documents part, it could be saved in your downloads. Just select that data and bring it here, and that's done without going into the hassle of get WD. Okay. Or and by the way, if you are still interested in setting the working directory, I am showing you here a very easy way to obtain working directory. So if you click here, click like this, and you see here it says C/users/ that is the admin, and then the folder where it is located in. Now the question is, there is a easy way to set or change the. So, but I find this a little bit tedious to do. So you can go directly to session, and then you can already see it says set working directory. and it says to source file location. So you can go here. Just a second. I think there is a problem here. So you can go to here and you can choose the directory. So you can choose here, let's say downloads. You can open it here. And now the directory is set to downloads. So you can write enter and then you write get WD this, and then you can check whether it is on downloads or not. I think it's taking a little bit more time. There you have it. So that's the setting of the working directory. So let's go to the actual thing here. So as you can see, I have written certain commands to plot the graph that we want to do. So I at least want to do a box plot, and I will also want to do a bar plot. But something more related to the stuff, as you can see here, if you click on tools, then what you can see here is there is a list of various kind of options. So you can see you can see here install packages, you can check for package updates, you can see version control, you can see terminal, you can see background jobs, add-ins, memory, and so on and so forth. So I think people who are using R on a daily basis, this is very important for them. So whenever you start R Studio, for instance, please click here and check for package updates. So this says that retrieving package installation context, and in a few moments, it will show you which packages are left to be installed and which packages need an installation that you can see. So the pop-up box has arrived, and I think it will show you the list of packages that are already there. That is a fascinating issue with my computer, although it's 3 years old, but I don't know, it takes a lot of time to process stuff. So these packages that you see here, and it shows that what are the new versions that could be available now. So like BTOPS, the latest version is available, and so on, and etc. I mean, for the time being, I am skipping this because anyway, during the sessions of today, we will install certain packages that are already here. So that's why I'm skipping this for a moment, but it is always advisable. People who are opening their computers daily, opening RStudio daily, please do update packages in a very regular interval. Okay, so we will do the plotting of a dummy data set. So we will directly go from here. So you can see that the first package that I need is called ggpubr, and I explained that before. And I have also called a package which is actually unnecessary in this case, but I have still called it because I will deal with an Excel sheet very soon. Sometimes what happens is if a package is already installed and you still don't know what is happening internally, day by day certain versions keep so the developers of these packages, they keep updating these versions. And sometimes it is a Herculean task to note which packages are updated, which packages need to be updated, etc. So it's all have to be done sometimes through trial and error. So you can click all this, and then it is called here. So as you can see, the console is working really fine, that if you call a function here, you can directly see that. So here comes an important terminology that is called data.frame. And as I explained to you in the slides, that data frame means it's a combination of many stuff. It's a combination of integers. It's a combination of information that is directly needed to plot a nice graph, that may be in terms of bar plot or maybe in terms of box plot. So if I call this function, and this is the variable that you are calling the function to, the name of the variable I have given df, you can put whatever name you want to give. There is a less than symbol here, and there is a hyphen symbol here. That means you are assigning this data dot frame to this one. That's the df stuff. So if you call it like this, so you can see in the environment that what do you see here? 180 observations of two variables, right? So if you click it here, maybe you can clip it here. I don't know, why does it take so much time? Okay. So this data that you see here has been taken with permission from Professor Ute Footnex. That's my supervisor currently. And I have created a dummy data set out of it just for the sake of this session, today's session, at least for session 1 and session 2. Session 3 and certain portions of session 2 we will do with the iris data set. But this I wanted to show you with a real life example. So essentially, so let's understand the structure of the data here. So JA, if somebody is here from the plant background, JA stands for jasmonate. Yeah, so it's a hormone. And treatment is that I have three kind of genotypes here. So I have something called as wild type, and something called as mutant, and also something called as overexpression. Yeah, and they have conditions. So if you see something with C, that means control. If you see something with D, that means drought. That's the stress. So, and in these conditions, the hormone JA has been, just a moment. Yeah, this has been shown, measured. So with this, I want you to show a box plot, a bar plot, and then a box plot. So here I decided that instead of manually putting colors to all the genotype, I will fix a color for the condition. So let me fix a color for condition that is the control. Let's say we have control as steel blue and drought as fire brick. Point to be noted here is a question which you guys can ask yourself, that okay, so direct blue color is available, direct red color is available, but I still chose steel blue and fire brick because in the European standard, blue and red color is considered to be colorblind colors. So people usually avoid it and go around something which is also red, also blue, but not the tinge that is there, or not the hue that is there in the red and blue, the traditional very dark red and very dark blue. That is avoided actually. So I have created a set of my own colors, and as you can see here also I have put a variable that is called my_colors. And then you have something which is here C, it's called C, and then you have a bracket open and a bracket closed, and within that I have called and I have assigned which genotype and which condition I call which function. Yeah. So let me call this function. In order to call some function, just select the function with the cursor and press Ctrl plus Enter. So it will be stored. So you see it now. It updated itself here. So it has a part with my colors, with steel blue and so on, with steel blue, fire brick, etc. And then with this, let's go to the bar plot. So as you can see, for the bar plot, I have used GG bar plot. Is the part of the function from the GG bar package. And so it has the following way to do it. So you have to open the bracket, then you have to call the variable in which your data was stored. So you just saw that we had stored the dummy data, and you are calling which is your x variable. So in our case, so the x variable was treatment, and the y variable was the jasmonate, that is JA. And here add equals to, I have put mean standard deviation that I want to show in the plot itself. And then I have put jitter, which I explained to you moments back. And I thought that I will also show what could be a possible variation to this. So I have chosen to separate the colors by treatment, and my palette is the custom colors that I created. And this is something called position dodge. So it's just to make it more beautiful, more appealing to the eyes while the plot is made. Yeah. So let's now do the plotting. So I have saved the bar plot in an additional variable, and I have called the variable at the end of creating the plot. Okay, so let us do it and see what errors are coming. Here comes the output. One of the reasons why sometimes plots take huge amount of time. Okay, so looks like there is a minor problem in the data loading actually. So I have to load the data again. Yeah. So looks like the appearance of the data. So it was shuffled in between the sets somehow. So let me call it again, and hopefully now it doesn't create a problem. Please bear with me and my internet speed, and let me check what it has created.
Sir, we can have a break of 5 to 10 minutes so that you can check.
Sorry?
Sabarna, we can have a break of 5 10 minutes so that
Okay.
you can check what kind of error it is showing.
Okay,
And
okay, yeah.
okay,
Mhm.
so it's
Yeah.
almost 1 hour. So
Okay.
we will take 10 minutes break and then we
Mhm.
will rejoin. So it's 11:57.
Okay,
So
sir.
exactly at 12, 12:07 we will rejoin.
Okay, sir.
Meanwhile, I will request all the participants to mark their attendance by writing your name in chat box. Okay, Sabarna? So
Yes,
आप
sir. 1 second. So it will resume at 12:10, right?
yeah, tell 10, 12, 10.
Okay. Okay, sir. Okay, sir.
Okay,
Okay, sir. Yeah.
okay. We are about to start.
Yeah.
Sabarna, can we start?
Yes, sir. Yes.
Okay, okay. We can start.
Okay. So I will start from where I left for the break. So apparently, it looks like the all the contents were working. It's just there are some false errors that are coming. So I guess you can see my screen, which I was talking about. Um, so with the bar plots, I will generate it again in front of you and quickly. It was just a false error that was coming. It's nothing to do with the data nor with the comments, but I don't know where from the error was coming. Yeah. So I hope you can see the screen. So if you see the screen now, I think now it is clear as a bar plot, and you can see here what I mean by this. You can see a standard error bar here, and around this bar, you can see like kind of a kind of a small infection kind of stuff. So these are called jitters. This actually show that what kind of data was there for the plot. But essentially, this is the plot. It shows that okay, this is lesser, and this is more actually. So that's what it shows here. And you can see that I have differentiated it with different colors. And I showed you on the onset that I had one control. Control means nothing was treated, and drought D means something was treated. So treated, I used a red color, and control, I used a blue color. And of course, now you know the command, and you can play around with it and take a look at how you can change them. And quickly, I will show you the box plot as well to inform you about the various differences that can be there between a plot and a box plot. Just one more minute and it will come. Just my screen view has some problems today. So some things are working and some things don't. Some things are not working. Anyway, I think I will shift to the PowerPoint because I can show you the box plot again, because it looks like it takes infinity before it reaches there. Um, yeah, I will switch to my PowerPoint. Yeah, so essentially it will create something like this. So that's a box plot actually. So it has this fat line here, the dark line here that is called a median line, as they say it. Okay, so that would be some of the stuff.
Meanwhile, Sabarna, sorry for
Yes.
interrupting.
Yeah.
As we can see, these are some beautiful graphs,
Yeah.
and those commands are very difficult to remember. And I think that it is next to impossible to remember all the commands. So
Yes.
it will be very useful for us if you can provide in the command list in the form of a Word file so that we can directly
Sure.
use that.
Sure. Mhm.
I will share it with my participant. So
Yeah.
they can use the same command for their data to get those beautiful bar diagram. Because I have seen this particularly this bar diagram showing the distribution pattern of the data on the
Mhm.
bar itself.
Yes.
So
Yes.
this is very new to me, although I am using the R since last two,
Hmm.
3 years.
Hmm.
But even I was not aware about this command. So if
Okay.
you can provide like the graph along with the actual command so that we
Okay.
can do that.
Okay. Okay. Sure, sir. I will. Definitely after today's session, or maybe like in 1-2 days, you will get a Word document from my side with today's comments. And as you can see now, sir, while talking to you, it worked. That's the box plot
Yeah.
that I was talking about. So yes, as you can see here, that it also shows, as I was talking about the box plot, the median line and etc. So I think these were the two that I wanted to discuss for the first session. So now I will switch back to the next session. I will switch to full screen now. In the next session, we are going to talk about a little bit about testing. So as you saw to the party, all the participants, my data set that what I am doing in the data set that I have a set of treatments and I have the values. Okay, so I have essentially what I have is I have names of the treatments, I have values. But I don't know that whether all the treatments are significant or they are not significant, and what does significant actually mean in this case. Yeah. So we are going to talk about certain stuff today. At least try to talk about them if it fits within the time span. But this should be the thing which is really, really relevant now. So the first thing is, of course, the classical t-test. Then also we're going to talk about false positives. We're going to talk a little bit about normal distribution, Mann-Whitney test, ANOVA, Duncan, and Tucky maybe I will skip for today. And then we will see a little bit on linear modeling, correlation, and regression analysis. So until...
Sorry for one more interruption.
Yes, sir.
Actually, the participant may not be aware about all these things. So I will request if you can focus mainly on t-test, ANOVA, normal distribution.
Yeah. Yes, sir. Sir, actually, I have written I have written a great explanation to all these. So I thought that maybe as a one-liner or as 1 or 2 sentences, I will explain it, and then it will be
great.
Yeah. So then it will be okay for the participants to understand. Okay, so I will slowly move on. So the first is the very basic t-test, as I think most of you should know that it has a null hypothesis which takes into consideration, and from that null hypothesis that the differences is not significant between that and based on that, if the p-value is greater than 0.05, we fail to reject the null hypothesis. Yeah. So that means if the p is greater than 0.05, p-value, that is the outcome of the t-test, is greater than 0.05, that means that there is no significant difference between your data sets. And only if p is less than 0.05, there is a significant difference between what you are comparing in your data. Yeah. So that is the crux of the t-test, as they say. So some more things, just quickly I will say. So first is one-tailed and two-tailed. So if you see a cricket match, everyone here, if you see a cricket match here and see when the captains, they toss a coin, right, at the start of the game, and the coin has 2 heads. So a one-tailed and two-tailed means 2 sides of the coin or 1 side of the coin. So let's take an example. So I have X here, I have Y here. Just take a look at X and just take a look at Y. So with a one-tailed test, you can only say that X is less than Y. Only one side you can say. But with a two-tailed test, you can say that Y is greater than X and X is less than Y. That's the 2 ways of explaining or interpreting your data in a very nutshell, so to say. Yeah. Now the question becomes, then when you do t-test, then what is the most accepted format of t-test? I think this is two-tailed. That is most accepted worldwide. Because that you have 2 ways to interpret your data. That's why it is more accepted. So I will not go into details, but let me tell you what is a raw p-value and what is a corrected or adjusted p-value. So the p-value that I was talking about in the first slide here, that p can be less than 0.05, yes, and p can be greater than 0.05. Now, in the less than 0.05, if a p is less, let's say a p between one of your comparisons is 0.009, yeah. So by just looking at it, you can say that okay, it is significant because it is 0.009. So it is less than 0.05, so it is significant. But statisticians say that this p-value might have false positives inside. And there are ways to correct this false positive. And this leads to something called a Bonferroni corrections or Benjamini-Hochberg method. And this leads to adjusted p-value. Some people write as PADJ. That is how it is written. These are the two kinds of p-value that are mostly used nowadays in statistical analysis. So that was with the t-test. And with the normalcy, so if you know what is a normal test, that is called Shapiro and Wilk, and it basically tests the normal distribution that comes from your mean of the data. So how your data is clustered and which points fit and etc. So in case of a normal distribution, what happens is the opposite. So if your p value is greater than 0.05, then it is normally distributed. Now, many people might wonder that sometimes what happens if normally distributed data is not there. Let's say but still you want to test certain methods of t-test. So this is called Mann-Whitney test. Yeah. So this is a way how to test when your data is not normally distributed. It can happen when you have very less samples. And for that, of course, the distribution will not be possible. So the next way to do statistics, because if your data is more complicated and it has more layers of treatments, or let's say more layers of comparison, let's say, then of course one might switch to ANOVA at that point, because ANOVA in that way it can test the effect of the variable in a very nice, let's say error-free way. And of course, ANOVA comes with another test that is called Duncan. And with the Duncan test, it leads to lettering. That's called, some letters are really generated. And I'll talk to that when I do that in front of you. It's very easily done. And we can see why ANOVA might be a superior technique as compared to the t-test. So as you see, there are many ways to check for significance, everyone here. It all depends, and in many forms it has been used in science mostly. But I guess the uniformity is what that matters. Like for instance, in biotechnology, I think if you see Duncan is mostly used and etc. But all the tests, if you know how to use that, you can use it uniformly. So if you work, for instance, people who are going to do at the end bachelor thesis as a form of research, I guess there's the new format here. And if you are planning to test your data with something, let's say you plan to test your data with Duncans, so please keep it Duncans. Don't change it to something else in the middle. That's the important thing that matters. Okay, so, and I will also talk a little bit about correlation, because that's what I planned also to show. So it's essentially the relationship between two variables. It could be a positive correlation, it could be a negative correlation, it could be a no correlation at all. And sometimes correlation is through heat maps like this. And it shows, so let's say the red colors show, the blue colors show negative correlation, and the red colors show negative correlation, and the red colors show positive correlation, and so on and so forth. So with this, I will very quickly, just right now, so we can start. Just right now, so we can start. So as you can see, so for instance, if your Excel sheet, and amount of hormone, no? But you want to insert another column or another row, you can do that with this package. That is, and of course, you have agricholine, that is for the t-test, for the ANOVA, and read Excel, big Excel sheets, and deep player, that is also another package that is used when you are very much using Excel sheets, and you are inserting columns here, rows here, etc. Okay, so with the t-test, this is the function here. You will get a full Word document from me that shows the data is the same that I just showed you guys before. So I will run the command. Sorry. So I think there was a problem. I think we are done. So yeah, I think you can see my screen. So that is the t-test which just happened. And let me show you very quickly what did it compare against. So you see, that was if you remember my data that I showed, that was jasmonate versus various line. And you have control as the first group and second as the second group as drought. So control where no treatment is there and drought where some treatment is there. Yeah? So, and you can see here. So you can see two columns, P and PADJ. Yeah, you can see P is 5.4 e raised to the power minus 39, and PADJ is also like that. So that means there was no false positive that was associated with this data, at least. Yeah, but there are cases when the data is huge and the situation leads to more false positives, then PADJ becomes slightly different from P. And then you have to consider your data based on PADJ only. Okay, so I will now return to my R sheet as we talked about. So we are going to do ANOVA as well. So we will do Dunkle multiple range testing, and I will show why it is so significant and why it is necessary. Okay, guys. So if you see here, what does it say in the output? So it says OEC as D, OED at A, WTC as E, WTD as B, mute C as DE, and mute D as C. Now, how to interpret these results? So this lettering is from low to high, from high to low. So A represents the highest amount of significance, this A. And D represents the lowest amount of significance. That's absolutely the lowest amount. Yeah. And
I think E.
Sorry, I'm very sorry, sir. E represents the lowest amount of significance, and something in the middle that represents that something which is significant, but if you consider D and DE, so DE is somehow lesser significant than D. And if you consider DE and E, so because one is different here, the D variable, D letter is different. So that means DE's significance is slightly higher than E, and so on and so forth. But the main thing, how to you have these in the always in that order. So here the lowest was E, and that's how you devise the various groups and understand what is the significant. But if you ask me, I think ANOVA is always superior to a t-test. So let's go with the Shapiro test results. And
This t-value huge.
which one, sir? This one.
Yeah, 11, 6,
No. These are the mean of the. These are the means. The mean point of the variable that I am testing. As I told that I am comparing my hormone amount in everything. Majority is a normal distribution with the Shapiro and Wilk test. So that means you can do t-test or ANOVA with this because most of the sets of the data are normally distributed in that case. Now, maybe I will maybe skip the Mann-Whitney for now, because I think I will share the commands at some point, because as you saw here that the data mostly is normal. So I think Mann-Whitney application will not be necessary right now, but although I made all these commands by myself and I pasted it here. And I will share this for the later stuff. So let me quickly show the correlation and regression. I have used the iris dataset for this purpose. So if I just do it, it's a very simple command, doesn't take much more time. And here is the correlation matrix that we need, and then we need to call a function that is called chordplot, and I will do that. And you can then make a nice graph out of it. Yeah, so you can see here that this graph, so if anybody is interested, so iris dataset, let me repeat, is from this plant which has the calculation of various variables: sepal length, sepal width, petal length, petal width, and then of course through this chord plot we try to understand which could be the correlation between what kind of values that can be correlated with one another, and what could be the how to interpret it. So here the pattern is a little bit different. So the more blue it is, the more positively correlated it is, and the more red it is, the more negatively correlated it is. So as you can see by the darker blue color, let's say between here and here, sepal length here and this one. And this is more positively correlated because you are comparing with the same variable. But if you see sepal length and sepal width, it's kind of this color, which is in the lower side of the panel, and that means kind of a negative correlation between the two. And if you see here with sepal length and petal length, if you see a kind of a blue color here, so that means it is kind of positively correlated. So if you want to create let's say regression, how they are related on a line, if you want to create the line and the fit line, so here is the commands, and I will do it for you in a second. So just to show you the scatter plot and how it should look like. And here they are comparing the relationship between sepal length and petal length. So here it is. So how to interpret this line? As if you remember my plot before in the introduction part, that all of these three, so in the iris dataset, there are three species: Setosa, versicolor, and virginica. Yeah, so the highest positive correlation was shown by virginica, as you can see with the very high line and with the kind of the plots. The second highest was shown by versicolor, and that was a little bit that was not that much profound. The line is not, so it is much lesser than the first line from the virginica. And the lowest is the Setosa, because it's not a perfect line with a perfect slope. Anyway, so the conclusion that comes is from here is that the highest correlated species in this case is Virginica. So that was it with the iris dataset, and I think let me show you another stuff here. So I also generated a linear model in case somebody is interested here. There is something called as R square, so 0.76. That means a huge positive correlation, but unfortunately doesn't differentiate between the species. So once again, repeating for people who are not comfortable with the iris dataset, iris dataset is a classically used dataset from 3 different species of iris. So iris setosa, iris versicolor, and iris virginica. And then it shows several parameters, and this dataset is widely used by statisticians to learn and simulate various graphs and various plots. So that was it. I think with this part at least of the session. So I think I will little bit pause here, sir, and open the floor for any questions that may come, if that is okay.
Yes, sir. But before we start on the question answer, I will
Yeah.
explain that all the data set what Sabarna is explaining, they are ready-made our studio, and you can directly download it. And classically, we are using these data set to practice, and based on this practice, we can use our data sets because there are readily available commands on Google, and with the expert like Sabarna, they can provide us. Otherwise, we can directly search for the command on Google. So, but sir, just before we start on the floor for question answer, I just want you to explain little bit about the Duncan test where you have explained A, B, C, D, E.
Mm-hmm.
So these are the things we get from various software like SPSS, PSPS, or like this R software.
Mm-hmm.
But can you also do manual calculation to find out, okay, this will comes under a group of A, then B or C?
That's a very good question, sir. Unfortunately, the answer is no. You
Oh,
can't. Unfortunately, the answer is no. I mean, theoretically, if you see it, it might sound to you like a little bit non-intuitive, because technically you can find ANOVA p-values for each group, and then okay, you can conclude that okay, maybe this is the highest significance, so it has to be A. But in that case, sir, if you are doing it manually, you cannot place which is the heterogeneous letter. So
yeah,
AB you can place, but what is CDE, that you
yeah.
cannot
And
place.
that was my question. Like I can place, I can differentiate A, B, C, D, E.
Yes,
But the moment it is B, C or B, C, D or C, D or C, D, E, then
yes.
it has become problem for me.
Yes, sir. Because I think, firstly, this is itself very tedious. The way it is assigned a CD, the way it is assigned a CDF, it becomes more tedious. And it's with the command, the algorithm that runs, that decides all this, and then you get the final output.
Okay, okay. And one more thing, like multiple correlation.
Yes.
So, uh, can you just show us your slide where you are showing the multiple correlation in the graph form, so that
Yes.
to those who are not aware what that graph is depicting, we can explain.
This one.
Sir, your slide is not visible.
Oh, my slide. Okay. Mhm. This one, I think.
No,
No.
so sorry,
Okay.
your screen is
Uh,
not sharing. You didn't share
okay.
it.
Oh, yeah. Sorry. I had turned it off. Now I think it should. I think you can see it now.
Yeah, now it's visible.
Yes,
Uh, no, the in our studio. Just
in
show
our
us
studio.
our
This one, or the heat map, you mean?
no, yeah, heat map, blue and
Oh,
orange color.
red. Yes. Mhm. Just a second. It's taking a lot of time
Yeah,
to
yeah.
load, sir.
This is the thing which actually fascinates me,
Yes, yes,
because like in Excel or in
yes.
many software, it's not possible for us to compare the correlation at the same time. But in R, it's become
Mhm.
very easy, like
Yes, sir.
length with
Yes.
sepal width or sepal length with
Yes.
petal length or petal width.
Yeah.
So we can, like here we are
Yeah.
using only four of them, but the same way I
Yeah.
think there is one more data which have the vehicle data in R.
Yeah.
So I just
Okay.
forgot the
Yeah.
name of the data. So if we have
Mhm.
10 different parameter like plant height, seed length, seed weight, leaf length, leaf weight, whatever parameter
Mhm.
we are having, we can compare all of them simultaneously in a single command.
Yeah.
Then all the participant must understand what this picture is depicting. This picture is depicting, I think there is some disturbance.
Somebody's microphone is not working.
Please, please. Jishan sir, can you find out? Okay, okay, thank you. So this is the fourth that if you will look at this picture, then you will understand the importance of bar. That in one command, how we can compare and find out the relationship between various parameter, and which is not easily possible to use other software like Excel. Might be in advanced form of Excel or advanced Excel it is available, but till date it's not easily available. We
Yes.
have to give separate command.
Yeah.
So this is one thing I want to share, and
Mhm.
yeah. So now we can open the floor for question for 5 minutes if they have any. Your hand or type your question in the chat box.
Yes.
No, I think there is no question. So, but meanwhile, we can take one more break.
Yes.
Yeah, 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 yeah.
Um.
Yeah, yeah. Sreshi
Mhm.
she's
Same
asking.
code.
Yeah, you
Mhm.
can read it.
Same code act differently sometimes in R. Yes, that is one thing that we have to be careful, and that puts immense pressure on somebody who is coding in R, because as I told, the people who have developed this algorithm, they update it every day, every hour. So you don't know which version you are using. 1 week back, something was working, the same command in same R, next week it doesn't work. Then you have to look for alternatives. This is, I think, one of the really bad cons of R, that there is a pro that it is very fast, but there is a con that it is very computer language dependent. So there are a lot of changes happening, and sometimes it is really difficult to understand where and why these changes come. So yes.
Any more question? You can raise your hand or just type.
Yes. 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 Mm-hmm. Mm-hmm. Mhm. That's a very good question, but the easy answer, sir, is no. R is completely free and universal. That's a very good feature in R. Unlike Python, I think some aspects of Python you need to buy a license. Something that is, I think you will get to know. I think maybe in the next session that is there for Python. I think that is called MATLAB, and MATLAB you need to buy a license for it. But I think for Art Studio is universal. It's free.
And okay, is there any dedicated website where we can get all the commands, updated commands? Means there are many websites which gives command, but like
Yeah.
Stacy raised the question, sometimes it's work, sometimes it don't work, and
Yeah.
every time we have to look for the same command in Google. Okay, okay, what is
Yes.
the right command?
Yes.
So
Exactly.
is there
That
any
is one.
dedicated
Mm,
website or link so that we can get the updated command? No.
No, sir. I think the one answer could be, let me help you in this regard. If you find problems that one command, last week it was working and same command is not working anymore, try to look at the error. Try to look at the error and try to understand the error message that comes at the end of the hour when something is not working. Try to understand that, and if you Google it accordingly, I think you will get a response why and how you could solve it.
But the problem is that the person like you who day to day work on the art, it's easier. But the person
Mhm.
like us who like once in a blue moon we open the art studio and start working on it, and then we could open our notes. Okay, this was the command. But
Mhm.
the moment we find that kind of problem, it's not easier. Even if we found the error, but we
Mhm.
are unable to fix it.
Mhm.
This
Um,
I'm asking.
Sir, let me give you an answer which maybe some people might find it unpleasant. I don't know whether I should say this or not. So, I mean, if you get some very difficult situations like this, you can approach ChatGPT, but point to be noted here, ChatGPT is language learning model. ChatGPT also at some point cannot solve your question because it's also learning from you. It's a language learning model. So I have tried this many times with my own students whom I supervise. So they also, so ChatGPT is also unable. It's something, it carves out something, you paste it there, it doesn't work because it doesn't know how to solve it. So, but ultimately, I think that the solution will be trial and error.
Yes, yes.
Yeah. Mm-hmm. Mm-hmm. Um, yeah. Sir, the answer is yes and no at the same time. Because, firstly, um, how we can identify that chat gpt makes error during that, you can only do that when you yourself have a very good knowledge of r that it is making an error here. That once happened to me when I was learning also r back then. So that was one thing, and second thing, look at your result. If the chat gpt's code can give you result what you need, then okay, it is working. But if it is showing unexpected erroneous messages or maybe bad plotting or bad graphics, then that means it has terribly failed with your specific question in R.
Okay, so with this, we are taking the second break, and I
Yeah.
think 10 minutes will be sufficient, sir.
Sir, hello,
Yes,
sir.
sir.
Actually, I have a query.
Just raise a question.
Yes, sir. Thank you, Sabarna, sir, for your informative session. So, I really wanted to ask, how can we resolve the issue encountered during the raster data exporting and analysis? Actually, I'm working on the raster data using R.
Okay.
So, yeah, that's
Mhm.
what these are my
What kind of problems that do you have during
Yeah,
exporting?
actually, sir, yeah, during exporting, I have a lot of why, while exporting the raster data in R, why they already taken the scaling, automatically scaling kind of things, and many we are unable to, create the fishnets and all these things. So, how can we like that kind of how can we understand ki which library and which function is applied to during that time, because you said these question, because same problem I'm facing कि last week ये work कर रहा था libraries, और next week it will not work out,
Mhm.
so I don't know
Mhm.
how
Mhm.
can we fix it, these problem while using.
Okay. So I think the problem that you are sharing is a very big, it's a big and more general problem. But the one solution could be that when you have, although I am not that much into raster data that you suggested, but about exporting, if you have so much problems, then I think one of the formats that don't create issues is TXT. That's the text format. You can give it a try, definitely. And if at all it doesn't work, I mean, take a look at where your error is happening, try to understand that, and then I think the approach or the solution will be more
Sir, actually, I'm exporting the raster data in TIFF format. So during the
okay. If
exporting time, the TIFF TIFF
okay,
format.
that's the image format.
Yeah,
Okay. Hmm.
yeah. Well, actually, my data is from. I had taken the data, the raster data from the Landsat. Okay,
Lancet.
so while for the LULC analysis and all these things. So
Hmm.
during that time. Most of the raster data are very very heavy data, so while during this exporting and during the data analysis, so there are a lot of raster packages and all, so I'm not able to understand how to resolve all these issue while it's coming the error like
Okay, okay, just give me a second, just give me a second.
Sir, you can, Sabarna,
Yeah.
you can answer
Yes.
after the break. Can we take
Okay.
break?
Yeah, sure. We can take break. Yeah.
So participant, Sabarna will answer your query after
yes
the
sir.
break.
Okay
Okay.
sir, okay,
So
no
we can
problem.
use this time for to
Yes sir,
take a
yes
break.
sure, take care.
Okay. All the participants are requested to write your name for the attendance. And today we are very short. So have patience and have some tea or coffee meanwhile.
Okay, sir.
Okay. So
Okay.
we will rejoin after exactly at 1 p.m.
10.
Yeah,
Oh, one,
1 5, 1 5.
1 5, yes. One
Yes,
five.
sir.
Yeah.
Okay, sir.
Okay.
Because it's Sunday, we don't want to waste. We have already wasted so many Sundays of our participants.
Yes, sir.
Yes.
Yeah.
So we will try to finish it as soon as possible.
Okay, okay. Yeah. Okay.
We are about to start.
Yes, sir.
So, can we start?
Yes, sir. Yes, sir. Okay.
Okay.
Okay. So before I start sharing my screen and go to the last session for today, the question that I stopped before in the last session. So the person who was asking the question about the raster dataset, just to complete the answer, that appears like it's a more broader question, and maybe it's specific to your own data set. So it could be only be solved that if I can get an overview of the exact problem. But for now, the solution that I can give you, that if you have problems in exporting the data set itself, then go to the very exporting the image itself, go to the very basic formats of images like PNG and JPEG. That might help. And otherwise, I think you have to share the problem directly to me, I think, and to my email address, and then we can get a look of the problem. That would be the answer. Okay. So moving on to the next session. That's the last session for today. Just a second. I will start sharing my screen. I hope you can see my screen now. Okay, so before I go to the last session, let me revise a little bit what we have been doing today to give you an proper flavor of the entire thing that we studied today. So we took a look at our data. We took a look at box plots. We took a look at bar plots. And we also took a look at how to show standard error, standard deviation. And we also learned about basic statistical tests and something advanced as well, because we also took a look at ANOVA and Duncan's test with the group letterings. And these are the certain things that we took a look at today. So I will end today's session with maybe two more very advanced statistical tools. And just to give you a brief highlight, and as this is something which people also do these days and might be relevant to people, especially who want to rigorously learn data science with R. So the first thing I will explain to you, don't get overwhelmed by the structure of the plot. So there is something called principal component. Yeah? So that is called PCA. So let us say that if you have a data set, you take any data set, could be the IRIS data set as well. So in the IRIS data set, just try to remember that what you had three species, and then you had data from petal length, sepal width, etc. Am I correct? So now the question that statisticians ask is that in this kind of data, what is the inevitable thing? What is the thing which is bound to occur from this data? So the answer is variance. There is a variable, there is a parameter that is called variance of your entire data set. So if you take the data set in whole, if you take the whole IRIS data set, you can ask the question that, okay, if the IRIS data set has X percentage of variance, where is the variance getting generated? That could be a question that can be asked in this aspect. Now, for this, statisticians came up with something called principal components. Essentially, there are two principal components. One is PC1 and one is PC2. This is how it is depicted. Sorry, I will change my phrasing. there are many principal components, but the most depicted one is PC1 versus PC2. It is only why, because these are the two components which show the most percentage of variance as compared to the other components. Yeah. So as you can see here, if you see this plot here, so let's say it has three different kind of parameters. You have rock fabric 1, rock fabric 2, rock fabric 3. See how nicely it has separated. Like there is a nice light blue color here, there is a nice medium blue color here, and there is a nice dark blue color here. Okay, so by plotting the variance as PC, principal component, as the name suggests, essentially these are variances. You can get a picture of your entire data set, how they are separated from one another. So here, the fabric 2 got separated from fabric 1 and fabric 3. Very nicely you can see. Albeit with some changes, like here there are some dark points in between. You can see that here also they are mixed in between. These could be the outliers. But in all, try to imagine the situation that I'm trying to focus here, that there is a nice separation of your data. And this is something which is demanded from researchers and from other people who use statistics from day to day, because this shows that your data is neat and clean. This shows that your data is homogeneous. So whenever there is a project, science project, scientific project or research project, let's say, and you target to cultivate a lot of data, then at the end, whether your data is homogeneous, whether your data could be trusted, whether your data has too much variance, whether your data is too much varying, these all answers can be answered simply by a principal component analysis plot. This will show how nicely your data, your different variables are separated. So if you remember the iris data set, there is sepal length, there is sepal width. I will show you an exemplary plot of the PCA for the iris dataset. Then you can understand what I am trying to say. That is one way that we can try to learn PCA. And the second way is that why and where it is applied. It is applied mostly in omics. People who do a lot of omics, I mean gene expression studies, but the meaning is still the same. You can see the different samples here, and you can see that every sample, every color from the other color. Can you see it or not? I think everybody can see it, that every color is nicely separated from the other color. There is no overlap. Although one might say that, okay, this blue color is a little bit closer to this, but in all, it is nicely separated. So that's what I'm trying to say, that if you encounter the term PCA at some point in analysis, or maybe at some point later in your research as well, this actually is a great tool to show how your data looks like actually, yeah, by creating this additional PC1 and PC2 components. That's how a PCA really works. And last thing for today, we'll of course look at the COM codes, and I will also do an exemplary PCA plot before finishing this session. So stay with me until then. And last but not the least, I will not put so much effort or put so much focus into it, because it is really advanced, and I can understand that it will be really difficult to grasp it. But this is the ultimate way to analyze the data. And this guy here, this gentleman here, this is called unsupervised machine learning. Now we are slowly going into machine learning algorithms. So this is something called k-means. So imagine the k-means algorithm works like if you are in a supermarket and you have bought a lot of stuff. No? You have bought apples, you have bought vegetables, you have bought, let's say you have bought banana. Then obviously when you pack it, then apples go into one, bananas go into one, the other stuff goes into one packet. It's something like that. So the computer also, the algorithm also does something like this. When you have a very huge data, by the way, I am all these during this session, I'm using the term huge many times, huge. In data science, n equals to 100 is actually nothing. N equals to 100 is usually nothing. Whatever we do below 100, n equals to below 100, that is not huge according to data science. So when n equals to 1000, 2000, 3000, then it becomes huge. And this kind of data do exist in biology. I mean, particularly people who deal with medical research, this kind of huge data do exist. And the more the huge data is, the more layered will be the way to interpret the data, to analyze the data, to explain the data. And k-means is one of that ways because it separates your data into different clusters. Yeah. So this is what I wanted to, I mean, just keep the various meticulous details with k-means. I just wanted to give you a flavor of that. So I will now go to my our screen once again. And we can take a look at a exemplary PCA plot, and I will use the IRIS dataset once again, so that we are on the same level. So I think you can see my screen now. Okay, so we are back to the R screen here. Okay, so we will do a PCA plot. So let me show. So I am using the IRIS data only. There is nothing new to it. I will do a very exemplary PCA plot for you so that you can see how it is done and how it looks like. It will take, I am afraid it will take 1 minute to load. It's a high resolution figure. Just 1 more minute. I think it will. It's a very high resolution figure. Takes a little bit more time. Let me run it again. So somehow, as you can see here, especially Sir, Sabinder sir, if you can see, sometimes there are some false error messages here. See, it says reached elapsed time limit. It did not say that there was error with the plot. It did not say that there was something really wrong. Just said that elapsed the time limit. But the moment I run it again, and now it works everything perfectly. So I guess, sir, that this is some of the things, some of the hurdles that one faces during use such programming languages.
Is it because of internet connectivity?
Sometimes, sir, but sometimes also it happens that R kind of gets confused itself between the packages.
Okay.
That's why the time limit also exceeds. So, yeah. So, so as everyone can see here, I have made a PCA plot out of the IRIS dataset. And as I told everybody that it has 2 axes. The first axis, everyone, it's called dimension 1, but it could be labeled as PC1 as I explained in the slide, and PC2, dimension 2, 22.9%. So you can see that the first PC, the first dimension here, Don't get overwhelmed if the term dimension comes. It's just a variable where the most variance, so 73% variance is stored. Dimension 2 is a variable where the lesser variance, 22.9% is stored. Okay. So, and these, when you make a plot like this shows that you see with, in case of Setosa, how the variables are nicely, I mean, without any kind of overlap with other species. Yeah, this kind of stuff really explains that the things that you see for Setosa is very unique, and this doesn't have an overlap with the other species. By the way, these are all species within the iris dataset. Yeah, you have the same parameters: petal length, petal width, sepal width, for three different species. And Setosa is the most unique one with almost no kind of sharing, because there is less, minimal or no overlap between the other species. If you look at versicolor, this is the yellow guy, and with the virginica, that is the gray guy. See, there is a significant amount of overlap. That means whatever properties there are in versicolor and virginica species, these are kind of shared and these are kind of overlapping, kind of similarity between the two. The last thing, if you see here, there is a term that is called biplot or biplot, let's say. Why it's a biplot? Because everyone, can you see there are 3 arrows here? Sorry, 4 arrows here. One here, one here, one here, and one here. So these are the contributing kind of arrows, contributing to the variance. So petal length, petal width, and sepal length contributes most to the variance as compared to sepal width. Something in that direction, because this is done when you have a very multi-layered data, and the data that I used for the iris dataset, it was indeed, it had many layers and many variables. And that's why sometimes what it is asked by the people is that which variance or the entire variance, as you can see here, is contributed by which variables. So these are the answers. That's the petal length, petal width, and sepal length. So I think with these small, with these two details, I think I will close the session for today, and I will invite for questions from everyone.
Sir, before anyone asks, I have few questions. Please, can you share your screen one more time?
Sure. Sure. I think you can see.
Yeah. So I have a question like this diagram is,
yes,
we are looking at 2D.
2D, yes.
Okay, but I think PCA is a 3D structure.
Not necessarily, sir. PCA is a n-dimensional structure. You can also create 4-dimensional PCA. So, sir, actually the thing what happens is when you calculate PCA, more than 12 dimensions are created.
Okay. So, but the pictures you are showing, is it 2D or 3D?
It is 2D.
Okay, if it is 2D, then it's okay. So the one thing is that we can interpret this figure in such a way that amongst Satuja and versicolor and virginica, out of these three, that if we will go for the phylogenetic tree, that Satuja will have a different clades compared to versicolor and virginica. Am I right?
Yes. Yes.
So the same kind of exercise we can perform with any kind of data?
Yes. Yes.
Okay. So that one more question from my side is that if we will share some data, like we have many of us may have some data, but just because of the unawareness and the complication about the handling of this PCA analysis, we are unable to frame any pictures like PCA, or even if we got this picture or plot, we are unable to interpret the data.
Okay.
So will it be okay for you if we will share our data so that you can not only analyze but also interpret the data so that we can get the information for publication?
Sure, sir. Should not be a problem.
Is it for me or for all the participant?
I think, sir, what you can do is, if I think you have my email ID.
Yes.
So I think, sir, you can share it with everyone, and from time to time, if I get the emails, I'll try to respond as much as I can.
Okay, so I will share your email ID with the participant,
Yeah.
and as we request in the beginning that please provide all the commands,
Yes, sir. Yes, sir.
updated command, because what I have in my PC, the command bank, but many of them is not working just because that they are outdated. As
Okay.
well as the data set, like you have used the iris data and which data only iris data, or there is some more data on which we can
Yes, exactly. So, sir, so for the correlation regression and for the PCA, I use the IRIS data, and for the t-test, box plot, bar plot, I use my dummy data set that I created.
Okay, will it be possible for you to
Sure,
share it with us?
sure, 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 sure.
You can change the head, the name of the head, so that no one can take your data or use your
No,
data.
no, no, sir. It's already dummyfied with the permission from my supervisor, so it should not be a problem.
Okay, okay.
Yeah,
So
yeah,
that's it from my side. Now, floor is so if anyone have any question, they can ask or
yeah,
type in chat box.
yeah. Yes, sir. Mm. That's a to prepare. That's a wonderful. Yeah, that's a wonderful question, sir. So, firstly, with the unpaid version of ChatGPT, you cannot do that. You have to have a paid version of ChatGPT or some more GPT. I think there is Gemini and the others, which are paid. So you have to get a license and then start. That's the first thing. And second thing, sir, I think I shared it with you in the last session. I think it requires human input sometimes. If it is really unknown, nobody knows, or there is no whereabouts of what could be happening in your data, then I think sometimes ChatGPT also fails. So I guess if you yourself have a good knowledge of R and then you start processing, you can take AI as your kind of help. But of course, sir, then it will lead to many errors. I have seen this situation many times, and particularly ChatGPT goes horribly wrong, or Gemini goes horribly wrong when it comes to this kind of complicated plotting. So there has to be some kind of human input behind that.
And there's one more thing which I want to ask in the same line of question.
Mhm.
So, when we are using these AI tools for preparing graphs and plots,
Mhm.
and if we are
Mhm.
using them for publication, what are the
Mhm.
ethical concerns? Do we have to acknowledge that we are using these AI tools to prepare the graph and images?
Mhm. So that's a very good and very relevant question, sir. I think it depends on the journal. Many journals, sir, by the now, including very top-notch journals like Nature Communications, Communications Biology, etc., they have accepted the fact that if you use ChatGPT or any AI as your helper, maybe in grammatical aspects or maybe improving a plot, it is well enough. It is acceptable, and you don't need to acknowledge that, because the opinion that they have it that if you use generative AI to create a project from scratch, I think a human will be able to recognize that, because that is depictable. On the other hand, if you have your own idea, you just use chat gpt or any kinds of AI just to be as a helper, okay, then that is acceptable, sir. And I think Elsevier and I guess Springer, they you have to acknowledge that you some journals of Springer, you have to acknowledge that you have used AI. Yeah, that is it. Hmm. Yes, sir.
Same query
Mm-hmm.
that when we are doing it, do we have to mention these in methodology also that we are using the this AI tool, or we are means using these um, message to get this information? Because if you change the frame of the language, it might get some different results. So do we have to do these things also?
Uh, I don't think so, sir. As I told you, sir, I mean, at the end of the day, I mean, people have started accepting that AI is a blessing of technology. At the end of the day, people have started acknowledging that fact. But yes, of course, AI usage comes with various limitations. And one of the limitations is this actually, what to mention, what not to mention. What I do is mostly, if you use AI only for improving stuff, there is not a need to acknowledge it at all. But of course, if your entire project is generated by AI and then you have improved it, then of course I think it's in LCV or some journals. You need to say that AI is an author. This is there.
Okay. So as you were showing the PCA analysis, heat map analysis,
Mhm.
so is it possible that we can use any other software which is not language based and we can still perform these analysis?
Yes, sir. I think there was already a session on that GraphPad.
Yeah, we had this session, but due to some reason we couldn't had it because of
Mm.
some personally uh,
Okay. Mhm.
engagements of the speaker. So we didn't had that session on GraphPad.
Mhm, mhm.
So okay, I did
Uh,
not know that we can do it on GraphPad also. Acha, one more
yes, sir.
thing.
It is definitely
Sorry, I
yes.
am having a lot of
Mm,
queries from you.
mm,
I hope it
mm.
doesn't bother
No,
you.
no,
So just one
no,
thing,
no, no.
just one last thing. So you know that there is one way of representation is geographical distribution. Can we do it in R also? Because nowadays it is even available through Excel also. So can we do this
Mhm.
even in R also? And how complicated could it be?
Mm, it is little complicated, sir, if you do it in R, because you have to clean your data. Because R sometimes, as I told many times, R gets confused with data frames, with data types. And I am pretty sure that if you're sharing mapping based data, then it's going to be complicated. And if it is complicated, then two packages there are in R. I think I showed it a little bit. This is called tidyverse. This comes into play, and the commands are really huge. Yes. Yeah.
Hmm. Okay, thanks for
Mm.
addressing all the queries. Over to you,
Yeah.
sir.
Thank you, sir.
Thank you, sir. And now floor is open for all the questions. You can raise your hand or
Yeah.
type your query in the chat box.
Mm-hmm.
So let's see. I think there is no query. It might be possible because many of them, for many of them, it is the first time they are getting the exposure of the R software,
Mm-hmm.
and it's not easy to understand the R software in one go.
Mm-hmm.
Although the way you have presented and the content you have included in your talk was extraordinary effort, but yes, it is a tough things to understand the complete R software in one go.
Yeah.
If they will practice it and finding any practical problem, they can share it with us or they can share it directly to you.
Yes.
This video will be readily available on YouTube,
Mm-hmm.
so we can share the YouTube link with you as well, so that if there
Yes,
is
sir.
any query, so
Yeah.
you can directly answer on the YouTube comment box.
Yes, sir. Definitely. Definitely.
So I think, sir, with this we can end today's session. Hmm? Done, sir? Okay, so now is the time for vote of thanks. So, on behalf of the organizing committee, I take this opportunity to express my heartfelt gratitude to all those who made this event in success. First and the foremost, we would like to extend our sincere gratitude to Sabarna Bhattacharya for his insightful and engaging lecture on the introduction to our software. It is a true privilege to see one of our own students share his expertise with us. Your clear explanation and practical example made a complex topic easy to understand, and we are incredibly proud of your accomplishment. Thank you once again for your valuable time and knowledge. I extend deep appreciation to Principal Sir Professor Narendra Singh, whose guidance and constant support made this program possible. To the IQAC coordinator, Professor P.K. Sisodia, and the bursar of the college, Professor Mukesh Jain, thank you for your constant support and guidance. I also acknowledge the constant effort of the convener SDCC, Dr. Saima, and fellow colleagues, Professor Dheeraj Singh, who ensured the smooth conduct of the session. A special thanks to all the participants who have registered and shown such enthusiastic engagement. Your presence is what gives meaning to this course. Last but not the least, thank you all once again for being with us. We look forward to continue learning, collaboration, and success throughout the course. So we'll see you all in the next and the last session of this course, and that is going to be very interesting because in there we will learn or we will get to know the basics of the another software which is Python. Thank you, Sabarna, and thank you everyone.
Thank you.
Have a nice day.
Yeah.
Bye bye.
Yeah.
And please make sure to pass attendance.
Okay. So you see you, sir. Sir, I will send you the codes in like 1 2 days.
Okay, okay, okay. Thank you. No,
Yeah.
no issue.
Okay. Sir. Yeah. Okay, sir. Okay,
Okay, bye
sir.
bye.
Bye, sir. Bye, sir.
Bye, everyone.
Bye.